Okay, good morning, everyone. Welcome to the class on autoimmunity. I hope you have gone through the PPT files that I shared a couple of days back. So, and I hope all of you are doing well. So, autoimmunity is divided into two parts, as I already um, discussed in the mail. The first part deals with immunological tolerance and its medical significance. The second part deals with what happens when immunological tolerance is broken, that is, autoimmune diseases. So let's start with the first lecture. So before coming to autoimmunity, which is essentially an immune response against our own antigens, against the self antigens. So before coming to that, we need to know about the normal immune response, which I'm sure all of you are aware, but let us cover it in a slight fast forward format. So today we'll essentially be covering this, focusing our attention on the adaptive immune response, because the innate immune response is non-specific, although innate immune response also discriminates between self and non-self. And but when we discuss autoimmunity, we are discussing an immune response against specific self antigens. So it is not a non-specific response. So it is mainly concerning the adaptive immunity. So now, as we know, what are the four cardinal features of adaptive immune response? That it is specific, that it is diverse, it has memory, and it has tolerance. So it acts against specific antigens only, as opposed to the innate immune, which targets non-specific. It has immense diversity. It has memory. What does that mean? That it responds with a faster, higher, and more efficient response on every subsequent exposure to the same antigen. So it remembers that antigen. So the second time when the antigen comes, the response is faster, it is higher, and is more effective, right? And tolerance. So the fourth characteristic was tolerance, that is this ability to discriminate between self and non-self. Steps of an adaptive immune response. So this is just an overview of what we already know. The first step is recognition of an antigen. Once the antigen has been recognized, B lymphocyte will recognize it using the B cell receptor, which is the membrane immunoglobulin, whereas T cell receptor T cells will recognize it using the T cell receptor. Then there will be activation of specific lymphocytes. So only those lymphocytes which have complementary receptors will get activated. Proliferation and differentiation. So there will be some exchange of signals. For example, in case of T cells, there will be co-receptors, there will be signal 2, which we'll see. So once those signals have been given, some interleukins will be released. They will also further increase the signal, and the cells will start proliferating. So once they start proliferating, they also will undergo some differentiation. So initially they were naive cells, but once they recognize the antigen and once they started proliferating and differentiating, they become either effector cells or memory cells. So this is known as clonal selection, clonal proliferation. Now the effector cells will go on to perform their function. They will eliminate their antigen, whereas the memory cells will stay back in the reservoir for any further episodes of the same infection. So elimination of the antigen will be followed by decline of the response. What does that mean? That the effector immune cells, maybe plasma cells in case of humoral immunity, or activated CD4, activated cytotoxic lymphocyte in case of cell-mediated immunity. So those will not survive. Okay, they will not stay. Once their work is done, they will leave. So how they will leave? They will die. So there will be programmed cell death or apoptosis, and there will be decline of the response. But your memory cells are long-lived naive cells. So they are nothing but they are naive cells, but they can survive for long. So they will continue to live on. So marriages made in heaven. I mean, I don't know how many of the man-made marriages are made in heaven, but this one, the lymphocyte and its antigen, it is definitely made in heaven. So it is not that the lymphocyte sees antigen and it's love at first sight. I mean, it is like that. So it is not like it has to do any dating or anything. Okay, it already knows that she is the one or he is the one. So lymphocytes are genetically pre-programmed to recognize a unique specific antigen. As I already said, B cells have the B cell receptor or the surface immunoglobulin or the membrane immunoglobulin. T cells have the T cell receptor, but these have been rearranged genetically even before the exposure of the antigen. So they already know, the naive cells, they know that their partner is somewhere. It has to be found. Okay, it may never find it. So not all, हर किसी को नहीं मिलता है यहाँ प्यार जिंदगी में. So not all naive T cells or not all naive B cells will be finding their antigen in their life. Like generations before us never found the coronavirus antigen, right? This one, the SARS coronavirus 2. But we are finding it. So our naive B cells or our naive T cells are lucky enough, or let's say unlucky enough, that they are able to, unlucky for us, that they are able to find their partner. So they will search. So just remember that their marriage is made in heaven. So multiple interactions help T cell activation. So this is your T cell activation. So you can see here that this is the T cell. It is the T cell receptor. So T cell receptor, like we already know, it recognizes the peptide only when it is complexed and presented with the MHC. So this is MHC class 2, because this is a CD4. If it is a CD4, then this is MHC class 2. If it is a CD8, then this is MHC class 1. Okay. CD4 and CD8. So this is this blue line that you see. This is your CD4 or your CD8, also known as the co-receptor. So this is the T cell receptor which binds to the peptide MHC complex. This is the co-receptor that recognizes the MHC class 1 or MHC class 2. And then there are co-stimulatory molecules. So I said that the T cell receptor avidity is not high enough. So you need other signals to support the T cell receptor peptide binding. So these other signals include, for example, CD28 molecule present on the T cell that recognizes the B7 on the antigen presenting cell. So this is the antigen presenting cell using the MHC class 1 or class 2 to present the peptide to the T cell receptor. But it also expresses certain other molecules known as the co-stimulatory molecules. So signal 1 is provided by the specific receptor antigen binding, but the signal 2 is provided by the co-stimulatory molecules like B7, also known as CD80 or CD86. Okay, B7-1 and B7-2, this 80 and 86. So I hope it is getting recorded. Yeah. So this is known as stimulation. This is known as co-stimulation. The role of innate immunity. So now you know that adaptive immune system is helped by the innate immune system. So how is it helped? First thing, obviously, you know that the macrophage is part of the innate immune system. That helps in endocytosing and processing the antigen and presenting it with the MHC. So antigen processing and presentation, that is an important role of the innate immune system. Or you can call it part of the adaptive as well, but the initiation of the adaptive immune response starts from from the innate immune system. But the second thing which we didn't discuss is that these co-stimulatory molecules like B7, so their regulation, their expression is upregulated by the immune response. So for example, when the toll-like receptor recognizes a particular bacterial antigen, and then it causes its endocytosis, it also sends a signal for certain cytokine production. And those cytokines will cause overexpression of the MHC, overexpression of the B7, so that now this antigen presenting cell is primed for its activity, primed for its recognition by the T lymphocyte. So you see that signal 1, okay, so I'm sorry. So the signal 1 is provided by the receptor binding, signal 2 is provided by the co-stimulatory molecule. So once the signal 1 and signal 2 have been given, so they will excite some genes. The most important one here is the interleukin 2 gene and the interleukin 2 receptor gene. So interleukin 2 acts like an autocrine. It's also paracrine, but here we are discussing autocrine signaling. So that interleukin 2 will sit on its own receptor. Okay, both the things are being produced here because of these. And once it sits on its own receptor, it will cause the cell cycle to get activated. So G1 to S shift or G2 to M checkpoint. So they will get activated and the T cell will undergo proliferation. So the naive T cells, so far naive, once it comes in contact with a specific antigen and it also receives a co-stimulatory signal, it starts undergoing proliferation and differentiation. Some of the clones, so this is now a clonal proliferation because a single cell is giving rise to the entire population. So some of cells of that clone are memory cells, they will stay back, rest are effector cells. Okay. So where does this antigen contact happen? So just remember that just as the lover is going around in search of his or her beloved, so where will he find him? Find her or him? It's not that he cannot go searching on the roads, he cannot go. So there are ways in which you meet your significant other, right? So you either contact them over, let's say you send Facebook friend requests, or you message her on Messenger or on WhatsApp, or there are some ways in which you will meet her in a cafe, meet him in a cafe or a restaurant or at some wedding. So there are specific locations at which these interactions happen. Likewise, in the body, the lymphocytes and the antigen interactions happen in the secondary lymphoid organs. So the primary lymphoid organs, bone marrow and thymus, are where they're produced. The secondary lymphoid organs, that is the lymph nodes and the spleen. Okay, lymph node for the lymphatics and spleen for the hematogenous blood. So these are the two destinations or the two platforms where the antigen and antibody, antigen and the receptor interaction takes place. So here you can see this is the lymph node with the afferent lymphatics. The dendritic cell has captured the antigen and is bringing it to the lymph node. Inside the lymph node, there are follicular B cell zones and T cell zones, so cortical zones and paracortical areas, right? So those areas is the place. So this is your T lymphocytes, okay, CD4 T lymphocytes. So they are waiting here in the So the cortex has the lymphoid follicles, which are the B cell areas, and the paracortex, which has T cell areas. So the CD4 lymphocyte comes in contact with the dendritic cell, presenting its specific antigen over here, and the interleukin signaling that we discussed happens. So it divides into a population. Similarly, the CD8 also so it can be either CD4 or CD8, depending upon which co-receptor it is expressing. Okay. So the proliferation takes place. So here you can see the naive cell receiving its signal 1. There will be signal 2 as well, which has not been shown. Interleukin 2 gene gets activated. Interleukin 2 receptor gene gets activated. Interleukin 2 acts in an autocrine fashion to cause T cell proliferation, some of which become effector cells, some of which stay behind, a minority, as memory cells. Okay, this can happen for both CD4 cells, which will recognize MHC class 1 peptide complex, or CD8 cell, MHC class 2 peptide complex, or CD8 cell, which will recognize MHC class 1 peptide complex. So now the differentiated effector and memory cells. So some memory cells can also go and home in other organs. Okay, but now we'll not discuss about memory cells much. So the effector cells will go, they will reach wherever there is the site. What they do depends. Like CD4 helper cells, what do they do? CD4 are known as helper cells because they will not directly do anything, but they will release certain other cytokines. So interleukin 2 we already discussed. They will release interleukin 3, interleukin 4, interleukin 5. Okay. So interleukin 3 acts on the bone marrow to cause further proliferation, but 4 and 5, they will cause differentiation of the B cell. So they will act on the B cell, they will help. So you have this CD4 effector cells, they will secrete cytokines. They will secrete cytokines, like I said, interleukin 4, 5, they will secrete cytokines like interferon gamma. So interferon gamma will act on the macrophage. It will activate the macrophage and produce killing. Similarly, it can also activate B lymphocytes. This has not been shown. So we'll see in the next slide. So it can activate the macrophages, it can activate the B cells, and it can induce killing using interleukins, growth factors called cytokines. And what does the cytotoxic lymphocyte do? It will directly perforate. It will directly perforate using perforins and granzymes. It will kill it. Also, the cytokines can recruit other inflammatory, so it can produce an inflammation, that is, it can recruit other leukocytes, other lymphocytes. It can act on further other T cells. So all that fashion it will do. Basically, just remember that CD4 will produce growth factors called cytokines, and it will activate macrophages, it will activate B lymphocytes, it can also activate the cytotoxic lymphocyte. And what the cytotoxic does, it does not secrete anything. It will just kill. It will kill the infected cell. So you have the naive helper T cell. Helper T cell gets activated by the CD4 co-receptor interactions. Also, there will be signal 2, like I said, CD28 interacting with B7. It can give rise to a different population. So we know that T helper cell secretes cytokines. So depending upon which cytokines it secretes, we have named them as T helper 1, T helper 2, T helper 17. So T helper 1 basically secretes interferon gamma. It helps the macrophages to kill the cells. T helper 2 will secrete interleukin 4, 5. It will help the B cells to make IgE, and it will also recruit eosinophils. So it helps deal with parasitic response. TH17, it helps, basically it brings the neutrophils and macrophages, so it helps the inflammatory response. So this is the antigen presenting cell. This is the naive T cell. This was the naive T cell. This is the activated T cell. This is the proliferated T effector cells, and this is the memory helper cell, memory T cell. So like I said, T helper 1 secretes interferon gamma, causes macrophage activation. Also, it can activate IgG production from B cells. So it helps in killing intracellular microbes. T helper 2 cell secretes interleukin 4, 5. It stimulates IgE production from the B lymphocytes. It can activate B cells. And it can activate eosinophils as well. So it helps dealing with parasites. And T helper 17 response, so interleukin 17, because of that the name comes, and it causes recruitment of neutrophils, monocytes. So it helps in dealing with extracellular bacteria, fungi, and all. So you already know that the hypersensitivity type 1 is mediated through the T helper 2. Because of it, there is IgE activation, IgE secretion, eosinophil activation, and that can cause mast cell release. So Dr. Swati already covered this in hypersensitivity type 1. Whereas hypersensitivity type 4, which is the cell mediated one, that is mainly by T helper 1 and T helper 17. Okay, so this is chronic inflammatory disease, like it has been said. This is allergy. So this is typical. Allergy is typically used to refer to type 1 hypersensitivity. And like I said, cytotoxic one, cytotoxic T cell. So this is your cytotoxic T cell. It recognizes using T cell receptor and CD8 protein. So CD8 will recognize the MHC class 1. T cell receptor will recognize the peptide MHC combination. It will also similarly get signal 2 from somewhere, and it will start getting clonally proliferating. Some will become memory cell. This is a memory. Most will become effector cell. That is the mature cytotoxic T lymphocyte. The mature cytotoxic T lymphocyte does not secrete growth factors like T helper cells. Like we saw, these were the growth factors, right? Cytokines. So all these were cytokines, but T cytotoxic does not secrete cytokines. It instead drills pores into the virus infected cell. So just understand that T helper cell is being presented by the antigen presenting cell with the antigen. Okay, they have purposefully endocytosed that antigen to present it to the helper cell. Whereas cytotoxic T cell is dealing with those cells which have become infected. So those cells never wanted to engulf the particular virus, or they got infected. So they are abnormal cells. They are not antigen presenting cells, but they are presenting cells because they have been infected. So they are altered self cells. Either they may be infected cells or they may be cancerous cells which are producing some abnormal antigen. So that cell is infected and is abnormal, is altered. So it has to be killed. So the T cell will kill it using something known as perforins which will drill pores, something known as granzymes. So this is okay. So lymphotoxin. So there are something known as granzymes also. So granzymes will induce apoptosis of it. Granzymes of fragmenting. So perforins will cause the cell lysis. Granzymes will cause cell apoptosis. Essentially, the cell will die. Okay. So like I said, CD4 helper cells deals with microbes that lives inside phagocytes. Microbes that can be killed by the phagocytes, macrophages. macrophages, for example, or even B lymphocytes. So phagocytes in ingested microbes in the vesicle. So CD4, so T helper, fine. Cytokine secretion, macrophage activation and killing, inflammation and killing of the microbes. Th17 brings forth your neutrophils and macrophages. Th1 activates them. Whereas cytotoxic, now they actually kill microbes that live inside the tissue cells. So it is not phagocytic cells. These are tissue cells which have become infected. So infected cell with the microbes or antigen. So this is the infected cell which is presenting its antigen to the cytotoxic T lymphocyte. It is asking to be killed. It is saying that, look, I'm infected, please kill me to save yourself, to save the body. So that is happening. Fine. Humoral immunity. So that was the cell-mediated immunity part. Humoral immunity is not, cell-mediated immunity essentially deals with ingested microbes, okay? Ingested, either purposefully ingested by the phagocytes or infected by the bacteria or whatever. So these are intracellular. Whereas humoral immunity deals with extracellular. So this is the B lymphocyte with its surface immunoglobulin. And these are your bacteria, fungi, whatever. And the B lymphocyte is recognizing them using their antibodies. So it is recognizing them. Once it has recognized, then what it does, it starts secreting those same antibodies. First, it recognized using a surface antibody, and then it starts secreting it. So these secreted antibodies are the effector molecules of humoral immunity. Just like cytokines are the effector molecules for helper CD4 cell-mediated immunity, the antibodies secreted ones are the effector molecules for the humoral immunity. So this essentially deals with, so this is mediated by antibodies and it deals with extracellular antigens like peptides, lipids, polysaccharides, okay? Now crosstalk. So just a moment. So the humoral immunity is helped in its task by the cell-mediated immune response. So we saw that the intracellular, now consider a particular bacteria. It has, some of its antigens are being recognized by the B lymphocyte directly. Whereas it has also been engulfed by the macrophage. It has been broken down and being presented in a proper way with the MHC to the T cell. So everything proceeds simultaneously. It's not like some antigens will only be recognized by B cells. So they are also being recognized by the T cell. The T helper clone gets activated and it starts secreting cytokines. Some of these cytokines reach the B lymphocyte and the B lymphocyte gets activated. So these such antigens are known as T-dependent antigens, thymus-dependent antigens, where the B cell response is guided by the T cell. So B cell will recognize it, but it will be unable to secrete antibodies. Okay, so far as until the T cell does not stimulate it. So when it comes in contact with the T helper cell, there is actual contact. Okay, it is known as the TB conjugate. So the T helper cell and the B cell, they contact each other, and the B cell gets some signals, and some cytokines will be secreted from T cell, like we already covered interleukin 4, interleukin 5, interferon gamma. So interferon gamma will cause the B cell to make IgG, or interleukin 4 will cause it to make IgE. So it depends which secreted antibody it will secrete. So just remember that once it receives those signals, it becomes a plasma cell, it starts secreting the antibody. So before the T cell has come into picture, it will not secrete. So this is the case with peptide antigens. So what we didn't cover is that the cell-mediated immunity mainly recognizes the peptide MHC complexes. So it only deals with peptide antigens. Whereas the humoral immunity can recognize not only peptides, it can also recognize your lipids, polysaccharides, lipopolysaccharides. So this, but for peptide antigens, it mainly needs the T cell to stimulate it. For the lipid, polysaccharide, lipopolysaccharide, it can directly mount a response, but for the peptide antigens, it needs the T cell help. Okay. So we'll, as we move, you will understand it better. So T dependent and T independent responses. So like I said, T dependent antigens are peptides mainly. So even though the surface antibody has recognized the peptide, that signal 1 is not enough to stimulate the B cell. It needs another signal 2 that comes when this same peptide activates a T helper cell, and that T helper cell now helps this B lymphocyte. How? So signal 1 is by the antibody, surface antibody, whereas signal 2 is by the CD40 and CD40 ligand interaction. So what is, again, just like in T cell we saw CD28 interacting with B7 molecules on the antigen presenting cell. Here there is something known as CD40, and the activated T helper cell expresses the CD40 ligand. So this interaction gives the signal 2. In case of a T independent antigen like the lipopolysaccharide, polysaccharide, lipid, there are several antibodies, surface antibodies. So that's no, in the antigen is polyvalent. So the antigen engages a lot of antibodies together, and they are sufficient to provide the two signals, signal 1 and signal 2. Okay, signal 2 is also received by some other molecules on the surface, but does not need the T cell help. So now we'll just see the classical response of a T dependent antigen. So you see that the antigen present starts with the cell mediated response. Okay. So antigen presenting cell presents the particular peptide to the T helper cell. Also the B7 and the CD28 co-stimulation happens. T helper cell gets activated. So activated T helper cell. Now on the other hand, your B lymphocyte has gotten attracted, has recognized its particular antigen using its own receptor. That we have not shown. But now this activated, but it is still waiting. Okay. So now activated T helper cell, once it comes in contact with this particular B cell, which has recognized its specific antigen, it is not going to bind to any cell. Okay. So this is a particular B lymphocyte that has already recognized its antigen. The T helper is only going to attach to that because the CD40 will be expressed by those B cells. CD40 ligand will be expressed by these. Similarly, in fact, this also starts expressing receptors for interleukin 4, for interferon gamma. So those are the ways in which the T cell will recognize the B lymphocyte and it will activate. So you can see the interleukin 4, interleukin 5, CD40, 40 ligand. All this causes the activation of B cell into plasma cells. These are the big factories of antibody production. They will start secreting the antibodies. Which kind of antibody? So one, it could be IgM. So which are the surface antibodies? Surface antibodies are IgM or IgD. Okay, those are the primary forms, IgM and IgD. Now once it has to secrete, the secreted IgM is the pentameric form which we discussed. We say that IgM is pentameric, but it is not pentameric on the surface. It is pentameric when it is secreted. So it can secrete IgM, or it may undergo class switching. Okay, the T helper cell, if it secretes interleukin 4, it induces class switching to IgG, to IgE. So instead of IgM, it can start secreting IgE. Similarly, if interferon gamma is being secreted, it can cause class switching to IgG. So we saw the Th1 and Th2. Th1 secretes interferon gamma, causes IgG production. Th2 secretes IL4, IL5, causes IgE production. So this is known as class switching or isotype switching. So just remember that the surface immunoglobulin are IgM and IgD, but when it gets activated into plasma cell, it has to secrete. So what it will secrete? It can secrete either IgM, okay, or it can secrete IgGA and E. So IgG under some T cell cytokine, IgE under some other T cell cytokine. It can also secrete IgA. So IL5, you can see here interleukin 5 causes IgA production. So IgA is a secreted antibody. So interleukin 4 is causing IgE, interleukin 5 is causing IgA. So IgA, we say that it is bivalent, right? If you remember the secreted. So this is produced in the GI secretions, the respiratory secretions, the GU secretions. This is the T-independent antigen. Like I said, it is polyvalent. So it engages multiple antibodies at the same time, which are sufficient to provide the signals. But just remember that T-independent response is not very efficient because it is not using the T helper cell. It can only secrete IgM. Okay, it cannot secrete IgG, A or E because that needs cytokine help. Second, it does not produce memory cells. So it only those cells get activated and they will just kill. So it is like that. So it is not very effective. That is why we say that peptide antigens are more effective, because peptide antigens undergo processing through the antigen processing pathway, and they get activated using the cell mediated immune response as well. So that is more effective because it ends up with you having memory cells. Whereas the T independent will produce, somewhat less of a response. It can only produce IgM. It cannot produce IgG, A and E. It does not have memory cells. The response will be overall less. So this is your naive B lymphocyte. Naive B lymphocyte with IgD and IgM. Okay, and it recognizes micro. It gets help from the specific T helper cells, proliferates and differentiates. So proliferation and differentiation. So either it can become plasma cell secreting IgM alone, it can become plasma cell secreting IgG or IgA or IgE. So those are known as class switching. And some of them, of course, will become memory cells. So they'll just stay back. And there is another phenomenon known as affinity maturation. So I won't go into the details, but this happens inside the lymphoid follicles. Inside the lymphoid follicles, some of the basically there is some genetic mutation still going on to find out that clone which will be the most effective against the antigen. So this is known as affinity maturation. So this happens under the influence of cytokines, that the slight small mutations keep happening so that the binding is perfected and the most perfect clone is selected for further growth. And finally, how the antibodies act, you already know. So antibodies can either coat the microbial surface and not allow it to act. This is known as neutralization. Or they can coat it and make it tastier for the phagocytes. So this is known as opsonization. Or they can fix the complement. Okay, so this is, or they can bind to cell and attract the natural killer cell. They can cause antibody dependent cell cytotoxicity. So this was something which your type 2 hypersensitivity. So these are all mechanisms involved in type 2 hypersensitivity, which Dr. Swati discussed. Okay, type 2 hypersensitivity is cytotoxic. So this first one was allergy, second was cytotoxic, acid, ACID, right? So that second cytotoxic, it acts through these pathways, or even the third one, immune complex mediated. So it can cause opsonization, phagocytosis. It can cause antibody dependent cell cytotoxicity. It can fix the complement. So it can activate the complement. So complement are basically glycoproteins which function in a cascade. So complement cascade will get activated. It will activate the membrane attack complex, which will kill the microbe. So neutralization, opsonization, complement activation, antibody dependent cell cytotoxicity. Complement can either directly kill, complement can also cause opsonization and phagocytosis. C3b. So C5 to C9 is the membrane attack complex, right? C5 to C9, C5, 6, 7, 8, 9. Whereas C3b is more of a opsonin. So C3b can also induce phagocytosis. So all of these are the effector mechanisms for humoral immunity. Now, how does it get deleted? Like I said, downregulation. So following elimination of pathogen, the immune system returns to homeostasis. Immune system meaning your effector cells. So majority of your effector cells will undergo apoptosis, but the long-lived memory cells will survive. Now, how are memory cells different from the naive cells? So they are nothing but an expanded pool of naive cells. So more numerous than the naive cells, and they respond faster and more effectively than the naive cells do. So they are like the naive cells, okay, but they express more receptors on the surface. So they will recognize the antigen faster, they can proliferate faster, and they are more numerous. So now you're finding your lover is easy. Once you have met the lover once, whatever, your girlfriend, boyfriend, the next time, of course, you can spot them from a crowd. It is not that difficult for you, right? So the first interaction is important. Second, subsequently, you develop an affinity and you can very well do it. So goal of vaccination is the generation of memory cells. So that is what we are trying to do now against the SARS coronavirus. We are trying to develop a vaccine so that once you have been vaccinated, you will have memory cells. Right now, everyone has naive cells. Okay. Right now, what are the weapons against coronavirus? You have your innate system and you have your naive cells. That's all. Those are the two things you have. Once your innate system is overpowered, which is happening in most of the cases, then the virus will enter your cells. So it will, in this case, it will enter your respiratory cells, the alveolar cells, and it will cause alveolar damage. It is causing interstitial, it is causing pneumonia, it is causing acute respiratory distress syndrome. So all of these things it is causing because your innate system is being overpowered. But if you had memory cells, you will be able to activate your CD8 lymphocytes, which will be able to kill those cells. So that is not happening right now. Okay. So cells of the adaptive immune system are the B lymphocyte and the T lymphocyte. T lymphocyte includes the helper cell population, the cytotoxic and the regulatory T cell population. B lymphocyte recognizes the microbe, gets activated into plasma cells, secretes various antibodies which act by neutralization or opsonization or complement activation. The T helper lymphocyte recognizes class 2 MHC complex peptide, receives co-stimulation, proliferates and differentiates into effector CD4 helper cell, which can secrete various cytokines. So cytokines can activate the macrophages like Th1 response. It can induce inflammation like the Th17 response. It can activate the other B cells, T cells, all those things it will do. It can induce class switching. Cytotoxic T lymphocyte recognizes the MHC class 1 complex peptide. So this is nothing but an infected cell. This was a phagocyte, which was deliberately consuming the antigen and presenting it to the T cell. This is not a phagocyte. This is not a warrior. This is basically a victim. Okay, it has been infected. So infected cell presenting the microbe is an altered self cell. Remember that term. Altered self cell, it could be either an infected cell, it could be a cancerous cell. So the cytotoxic T lymphocyte recognizes the MHC complex 1 peptide on the infected cell, kills it. Finished. Okay, it will release perforins, it will release granzymes, it will not secrete any cytokines or anything like the CD helper cell, it will simply kill it. Regulatory T cell. So this is an important part that we'll be covering in the immune tolerance. So regulatory T lymphocyte is simply something known, it suppresses the immune response. They were initially known as T suppressor cells. Okay, when they were first recognized, we thought that they were a subset of CD8 positive lymphocytes. But today we know that they are a subset of CD4 positive lymphocytes. So they are CD4 cells overexpressing some other molecules. Okay, so these are CD4 cells which help in suppressing the immune response, preventing excessive immune activation, the suppression of the immune response. So they will not allow the helper or the cytotoxic T lymphocyte, the naive helper and cytotoxic T lymphocyte to get proliferated. So they will suppress the immune response. Okay. Okay, I will stop the video right now.